bioequivalence and bioavailability studies. She also has extensive experience using knowledge management techniques to support CDER's regulatory information infrastructure. As a member of CDER's biomarker qualification program, she contributed towards guidance and policy development of biomarkers as a drug development tool, or DT. It is my pleasure to introduce Dr. Sanyal.
Good afternoon, everyone. I hope you all had a good lunch break and you are ready for the post-lunch session of this webinar. In this session, we will go through some more case studies. I'm sure you got some insight about clinical case studies from the previous presentation. During this presentation, I'm going to present a number of analytical BA/BE case studies. Before I start my talk, I need to add this disclaimer that this presentation reflects the views of the author and that it should not be construed to represent FDA's views or policies. For this part of the webinar, the learning objectives are understanding when rejection of an analytical run is justified, understanding the importance of complete and contemporaneous documentation during an analytical BA/BE study, and finally, we will discuss the importance of maintaining analytical run data and audit trails. For reference throughout this presentation, I will refer bioavailability and bioequivalence as BA/BE. In this presentation, I will be presenting 3 case studies. After presenting the first case study, I will ask a few questions to the audience and discuss answers. This pattern will be repeated for all 3 case studies. At the end of the presentation, I hope we will have some time to discuss additional questions from the audience. Okay, let's start the first case. In this one, we will discuss when it is justified to reject an analytical run. Consider the situation. During a method validation experiment, one precision and accuracy run was excluded from overall precision and accuracy statistical analysis. The method validation report stated that the precision and accuracy run did not meet the acceptance criteria since both high QC samples in that run failed. The incident was documented in an incident report and assessed as an assignable cause. Based on that, the precision and accuracy run was rejected. Although I would like to note that the exact nature of the assignable cause was not specified in the incident report. During inspection, upon questioning, the firm said that they suspected there were some sample processing error with samples used in the rejected precision and accuracy run. We should note that the method validation SOP specified that the precision and accuracy batches cannot be rejected without an assignable cause. So just having suspicion without any contemporaneous documentation is not good enough reason to reject a precision and accuracy run. Additionally, according to the 2018 bioanalytical method validation guidance, there are no acceptance criteria for QCs in a precision and accuracy run. So in realities, QCs cannot fail in such run. Overall, the point I want to make is that you need to justify and substantiate the cause based on which a run is being rejected in method validation experiment in an analytical BE study. Examples of assignable cause. Let's say the HPLC or the spectrometric apparatus malfunctions. There is some kind of electrical or mechanical issues such as the pump stopped or it's leaking. There is a power failure and so on. There might be a computer or network issues where the run was not recorded or data was not saved. And error during sample preparation may be counted as an assignable cause, provided the exact nature of the error is known and there is documentation available to substantiate the claim. In general, for all assignable cause claim to reject a run, you need to provide contemporaneous documentation and justification to support your claim. So that's the first case study, and as I mentioned in the beginning, we will go through a couple of questions and answers after each case study. Let's discuss some questions and answers relevant to case study 1. Good afternoon, everyone. I think now we will discuss some questions related to case study 1. So let's have the first question. The question is, during a precision and accuracy run in a method validation experiment for an analytical BE study, the auto-injector in the HPLC stopped working and incident report was filed and the precision and accuracy batch was rejected. The options for answers are rejection of that precision and accuracy batch is justified, and B, the precision and accuracy batch should not be rejected. I think we can wait for a few seconds and see if there's answers coming in the chat box. Yeah, I see some A's and B's. It's a mixed bag. Yeah, more A's than B for sure. Okay, so most of you have guessed it rightly. The correct answer in this case is A. So in this case, the rejection of the precision and accuracy batch is justified. As we just discussed in the first case study, failure of an analytical instrument while you are running the experiment can be designated as an assignable cause, provided you have contemporaneous documentation. Since in this case, we have some contemporaneous incident report filed, the rejection of the analytical run will be justified. Okay, moving on to the next question. The question is, 3 out of 6 non-zero calibrators failed to meet the acceptance criteria in a method validation run, and the run was rejected. The option for answers are A, rejection of the run is justified, and B, the run should not be rejected. Again, I'm looking at the chat box, and yes, I see most overwhelming option A, and that's the right answer. So if 3 out of 6 non-zero calibrators fail to meet the acceptance criteria in a method validation run, rejection of the run is justified. As we know from 2018 method validation guidance, bioanalytical method validation guidance states that the 75% and a minimum of 6 non-zero calibrators should meet the acceptance criteria in each validation run. Considering that if 3 out of 6 non-zero calibrators fail to meet the acceptance criteria, that validation run may be rejected. Okay, I think we can move to the next question. The question is, during a precision and analysis run in a method validation experiment, 2 out of 6 LQC samples showed more than 15% deviation from the nominal concentration. Those LQC values were excluded from the overall intra-run precision and accuracy calculation. Now, what do you think about the following statement? Is it true or false? And the statement is, all LQC samples should be included in the overall statistical analysis. I'm seeing again more true and some false. So let's discuss this. What should be done here? Should the LQC values be excluded or it should be considered? If you remember, I had one of the same scenario in case study 1, and the scenario was that a farm did not include HQC samples in the statistical analysis for precision and accuracy calculation. And the reason they provided was that HQC samples did not meet acceptance criteria. I want to point out again that according to 2018 bioanalytical method validation guidance, there is no acceptance criteria for QCs in a precision and accuracy run. Therefore, all QC samples should be considered for overall statistical analysis for precision and accuracy determination. And I think yesterday this issue was covered in some of the presentation. So just again, remember that for QCs and in a precision and accuracy run, there is no acceptance criteria. Whatever value you get, even if it's, there is more than 15% variation from the nominal concentration, those values should be included. I think that concludes the knowledge check for case study 1, and we will continue to case study 2 now. With that, we will now proceed to the second case study and discuss the importance of complete and contemporaneous documentation during an analytical BA/BE study. Let's consider this example. During an inspection, documentation regarding sample preparation was requested, but the firm could not provide complete documentation of preparing quality control samples used in long-term stability studies. Therefore, long-term stability studies could not be reconstructed. In another inspection, it was found that one set of subject samples was classified as run failure, therefore that set was rejected. Those samples were reprocessed and reanalyzed. When the reprocessing of samples was questioned, the firm stated that there was a suspected sample processing error, but there was no supporting evidence to substantiate the claim of sample processing error. So overall, I want to reiterate the importance of complete and contemporaneous documentation. In an analytical study, there may have been incidents either during method validation phase or sample analysis, and you may need to re-inject, reanalyze, or reprocess samples, but you should provide complete and contemporaneous documentation so that your reanalysis or reprocessing claim can be justified. With that, we will now discuss some questions related to the second case study. So we will be continuing with some questions with the second case study. And I'm also looking at some of your comments in the chat box. And I think at the end of the presentation, we can discuss some of those comments and questions. So question number 4, and the first question related to the second case study. The question is, during sample preparation in an analytical BE study, amount of buffer added to a set of subject samples was not correct due to the erroneous setting of the micropipetter. The analyst immediately caught the mistake, documented in the incident report, and that sample set was reprocessed. The options for answer are the course of action taken is justified, and B, the course of action taken is not justified. Okay, I'm seeing overwhelming A, and yes, you are all of you are saying A. That's right. The options A is the right answer here. The course of action taken is justified. Now, during analytical study, if there is any incident which may affect the results of a study, and we know, incidents happen. A batch or a run can be rejected provided there is contemporaneous documentation to justify the rejection. In this case, as we can see that there was an incident which was immediately caught by the analyst, a report was filed, and therefore reprocessing of this sample set is justified. Okay, moving on to the next question related to case study 2. The question is, during sample analysis in an analytical B study, a set of subject sample was reprocessed due to sample processing error, and the run was repeated. But there was no citation of the error in the source documentation. With that in mind, what do you think about the following statement? Is it true or false? And the statement is, to justify the repeat run, the error citation should be included in the source documentation. I'm seeing mostly true answers, and yes, that's the right answer here. So most of you are guessing it correctly. As the second case study examples showed, complete and contemporaneous documentation is really important, and it is an essential practice to an analytical B study if that study is submitted to a regulatory authority. A good documentation practice will save a lot of time during an audit. Unless proper documentation is available, it is hard to trace back sequence of events during the study conduct. Yesterday, Dr. Viswas elaborated the importance of proper documentation for analytical B studies. This morning, we heard from Dr. Pham and from many others that how important the proper documentation is whenever you are conducting a study, and it's to be submitted to a regulatory authority. So I want to put an emphasis again on having complete and contemporaneous documentation, which meets FDA ALCOA standard, and the ALCOA is an acronym for the phrase attributable, legible, contemporaneous, original, and accurate. I saw and comment here, don't you require an approved protocol for reprocessing? Yes, your protocol or SOP should clearly state in which, when you should reprocess your samples. But what I want to make the point here is that, let's say there is an incident where the reprocessing is warranted, but even then you should have proper documentation to make sure you write down everything what happened and the reason why you are reprocessing the samples. Another question, if the run was repeated correctly, is the purpose of the error citation only for contemporaneous purpose? So yes, if you repeat the run, then during an audit, there will be the question that why was the run repeated? And until you have complete and contemporaneous documentation, you won't be able to give the justification that why the run was repeated. So even if, as I just mentioned, even if it's approved by your SOP, you still should have contemporaneous documentation so that the study can be reconstructed. So whenever there is a question that why this run was repeated, you have the written report why. Okay, I think we can move to the next question here. And the question is, it's the last question related to case study 2. During an inspection, it was found that one of the -80 freezer containing subject samples stopped working due to power outage. The subject samples were temporarily moved to another -80 freezer, but the farm could not provide temperature records for the new freezer. Upon questioning, the farm explained that it was a temporary move and the subject samples were stored in the new freezer only for a week. Therefore, keeping temperature records of the new freezer would not be necessary. So the options for answers are: the farm should keep temperature records for all freezers, which is A, where subject samples were stored, and B, the temperature records for the freezer where samples were stored temporarily may not be needed to preserve. And I'm seeing overwhelming A in the chat box, and that's the right answer. Uh, the all the temperature records of all freezers, wherever subjects, chemicals, or standards, QCs, anything related to the study material was stored, everything should be preserved. So in this case, the temperature record for the freezer where the subject samples were stored temporarily should be preserved. And again, the point here is that proper documentation so that you can reconstruct the study. So in this case, if you have a gap between of the temperature records of subject samples, and during the audit, if you are asked that how do you know that the subject samples were stable, or nothing went wrong with the subject samples, to reconstruct that or to answer that question, you need the complete record, complete temperature records of all the freezers. So that's why it's really important to keep the records of all your incidents, as well as like this type of temperature records and so on. So I see some of the comments, and yeah, those are really good one. Irrespective of duration, all records should be documented. That's I saw another question. It says, why stating should rather than required? Now, what I will say that in OSIS, when we do the inspection, this is a scientific inspection rather. So if you look at the regulation, it's not specified exactly like that in the regulation. So during our inspection, all we would like to see that whether these studies can be reconstructed properly. Now, if there is any gaps in the record, that's where we question and we ask for the records, the total records. I'm looking at more comments. One question I see here: if there is no records of the new freezer, all the samples data are still accepted. I would say it depends. Now, let's say you don't have the records of the new freezer and you ran some analysis with that set of subject samples, moved it back to your old freezer, then we may ask for taste to you can, where you may need to show that the subject samples didn't go bad, or it was still there. So you may have to show some proof about that. Now, how are you going to do that? That's there may be other options, but I think the straightforward way in this case is just to preserve all the records, so that there isn't any questions during audit which comes up, and you may not have complete records to uh, show that everything was fine and nothing went wrong. Okay, I think this was the last question for case study 2. And now we will continue to case study 3. But please keep the chats coming. Let's proceed to the last case study of this presentation. In this one, I will provide some examples regarding best practices for retaining analytical study data. During an inspection, while checking the audit trails of an analytical run, it was found that the changes in peak integration parameter was not uniform. The peak integration parameter for different samples were applied differently. In another instance, it was found that one of the results table in one run was overwritten. The original version of the results table was not saved. It was also found that the sample type was changed to unknown from quality control after the analysis was completed, as you may see in this screenshot provided here. All these examples are not considered as the best practices to retain analytical study data. Peak integration parameters should be uniformly applied to all samples in the study. If the results table are overwritten for a reason, it should be justified and each of the original version of the results table should be preserved. New version of the results table should be saved with a new name. Additionally, sample type should not be changed once analysis is completed. Sometimes sample name or type may be changed before analysis, but that should not be the case once analysis is done. Now, let us discuss some questions related to case study 3. Okay, so we are back to discuss some questions related to case study 3. The first question: during an inspection, the results table audit trail showed the sample type was changed from quality control to unknown after completion of sample analysis. Is this an example of acceptable best practice to retain analytical study data? And the answers are yes or no. So yes, I'm seeing overwhelming no and B options, and you are right. This is not at all an acceptable practice to retain analytical study data. If you remember in the case study presented, the case study 3, there was an example where we have seen the audit trails showed that the sample type was changed from quality control to unknown after the analysis was completed. So that's total no. That should not happen. Sometimes the sample name or type may be changed in some cases before the analysis, but that should never be the case once data analysis are completed. Moving on to the next question. During a review of the chromatograms of an analytical run, the analyst changes the integration parameter for one sample but does not apply it for other samples throughout the run. The question is this an example of best practices to retain study data? The options are yes and B, no. So again, you have guessed it rightly that the obvious answer is B. The peak integration parameter should be uniformly applied to all samples in the study. Picking and choosing specific chromatograms for reintegration to make the study look good is not considered at all best practices to retain analytical study data. And there should be a protocol or SOP that when you can manually integrate a chromatogram or a peak, and that should be followed. And if it is changed, there also should be documentation available to show that. I have looking at the chat box and the answers are still coming, which is B. And again, as most of you have, almost all of you have guessed it rightly, the answer is B. And that concludes the knowledge check for case study 3. To sum up this presentation, I want to make a couple of points. First of all, it is highly recommended to look at the 2018 bioanalytical method validation guidance and the recommendations provided in that guidance while conducting an analytical BA/BE study. Regarding all case studies we discussed today, I would like to summarize that number one, with any unexpected events which may happen while conducting an analytical study or deviations from protocols, repeat analysis, all of these events should be supported by complete and contemporaneous documentation. Number two, rejecting a run or a sample should be justified and substantiated by proper documentation. And number three, think about the best practices for retaining study data. One way to do that is to maintain original and reintegrated or repeat data, and it should be supported by your firm's SOPs, protocols, or other written procedures. I have listed some FDA guidances for your reference. Chances are that you are already aware of these guidances, but I have listed them here so that this will be available in your slide deck. With that, I would like to conclude this presentation. I want to acknowledge and thank my colleagues who advised me to prepare this presentation. And I want to thank you all for your time. I hope you will enjoy other presentations we have for you this afternoon. And I will stop here and get back to the host. Thank you again. Okay, I see lots of good questions and comments coming in the chat box. Just they are going really fast, but I will try to answer some of them. So one question I saw about GLP. I think it was during my second case study presentation. So just wanted to mention that Part 58 GLP regulations do not apply to data from the human clinical trials. So what we are presenting here right now, the BAB case, the bioequivalent or bioavailability case studies, and that's a human clinical trial, and the GLP regulations is not applicable to this. Regarding manual integration of chromatograms or a peak, the farm or you should have written procedures when and how and which condition the peaks should be reintegrated manually. So that's the first thing. And also, as I said, that yes, I saw a comment that sometimes this automated integration takes longer. So, or to refine, you can manually integrate, but it should happen according to your farm's SOP, and there should be documentation to show that when and how you have reintegrated those pigs. One question regarding if the laboratory SOP for method validation sets acceptance criteria. So that's it is related to case study 1, I think. So the question is if the laboratory SOP for method validation sets acceptance criteria for QC values, even if the 2018 guidance do not contain any, shouldn't the lab follow their SOP or more restrictive than the guidance for the run acceptability? So I guess precision and accuracy run, your QC values are your QC values during method validation. So you are trying to validate the method. Um, in this case, first of all, if let's say some of the values are really skewed or it doesn't meet your acceptance criteria of your lab protocol, um, there might be you can do some inter-run statistical analysis with and without outliers. But overall, what I would say that in a precision and accuracy run, you have to include all of those runs, even if they are outliers. I mean, you can have calculations without them, that's fine, but there should be at least one calculation, overall calculation, statistical analysis that should include all those QC values. And again, you can have it without the outliers. One question regarding the draft guidance, FDA, ICH, M10 guidance. So this came up a couple of times yesterday and also today, this morning. But I would say that ICH M10 guidance, I know the comments period's closed, but it hasn't been finalized yet. And also FDA hasn't finalized or endorsed that guidance yet. I'm not sure when it will be finalized. So all we can say right now, our guiding principle is based on the 2018 method validation guidance. So that's the one we are following till now. One question is, but if the sample type is wrongly selected, I think that's related to case study 3 or the audit trail. And if it's realized during the result table saving, can we change the sample type with justification? I would say again it depends. So the rule is once you finalized analysis, there shouldn't be any change to the results table. Now, if it was wrongly selected and if you have contemporaneous documentation, I think it might be justified, but the rule again is not to change anything after the analysis is complete. I see some of other questions coming. One question says, if there were impurities in blank sample, then this impurity should be reduced in detection sample when doing equipment cleaning quantification. I am not exactly sure, but if yes, if there were impurities in blank samples, then I mean, once you realize that there were impurities in blank samples, so yeah, you should take all the measures you can. I think you can prepare fresh samples or you can clean up your instrument as required. So that's definitely according to your SOP. You should proceed with that. Okay, I think we can stop here and get ready for the next case study. And there were really some nice questions and comments. I really appreciate your participation and taking time through this presentation. And I would like to thank you all. And I will get back to Nora for the next presentation. Thank you again.
Thank you, Dr. Sanyal, for that great presentation. And again, thank you to the audience for the great engagement as well. We